Hi everybody, this is Mitch Kovacs, and the paper I'm presenting is called Adrenal fasciculata cells express T-type in rapidly and slowly activating L-type Ca2 plus channels that regulate cortisol secretion. It's by John J. Enyeart and Judith A. Enyeart. They're from the Department of Neuroscience at Wexner Medical Center in the Ohio State University at Columbus, Ohio. This paper was published in the American Journal of Physiology Cell Physiology this past March. Um, so that is a mouthful of a title, but the base level of understanding that you need before we get into this is that they are going to be studying cortisol. Um, so what's the big deal with cortisol? We learned in class that cortisol is pretty crucial for metabolism. It regulates the metabolism of most of the cells in the body. If you remember this table, it shows that cortisol comes from the adrenal gland, specifically the zona fasciculata of the adrenal cortex. And we learned about the regulation of that, so let's review the mechanism of regulation. So the primary control comes from the hypothalamus. The hypothalamus releases corticotropin releasing hormone, and that stimulates the pituitary to release another hormone called adrenocorticotropic hormone, or ACTH. That ACTH then stimulates the adrenal zona fasciculata, or AZF, as I've abbreviated here. The AZF can then release the cortisol, and that goes all over the body to the target cells. So this paper specifically is going to be looking at the mechanism of this step here. So we know about the control and the regulation, but we never actually learned about the mechanism itself of how ACTH can come in and then cause AZF to release cortisol. So what's the mechanism leading to the release of that? And you might be delighted to learn that we've actually gone over that mechanism before, and it's this. This is a G protein coupled receptor with cyclic AMP as a secondary messenger, and we should all know that by heart. But I'm going to review it again. So ACTH is the hormone. He comes in, binds to the receptor. The G protein, adenylyl cyclase, can convert ATP in the cell, specifically the AZF cell in the adrenal gland. That ATP is converted into cyclic AMP, which can then do a lot of stuff. One of the functions it has is to open ion channels. Specifically, in this case, we're going to see that it'll work on calcium channels. More of that in a little bit. So that was the mechanism. But what they didn't teach you is that there's actually another mechanism that can regulate AZF to release cortisol. And it's not just ACTH. There's this other hormone called angiotensin 2, or ANG2. And control from that is a little bit more complicated. There are precursors from the liver, and there's a bunch of steps in between there to get from those precursors to actually getting angiotensin 2. But that's not important. What you need to know is that angiotensin 2 similarly stimulates AZF cells in the adrenal cortex to release cortisol. But what's the mechanism of that? You'll be delighted to learn again that it's basically the same thing. We learned it at the same time. It's another G protein coupled receptor. This time it's specifically the IP3 DAG pathway. So angiotensin 2 is the hormone. He comes in and he activates he binds to the receptor, which can then activate the G protein. And that G protein, as we learned, stimulates the release of DAG and IP3. And IP3 can cause opening of calcium channels in the membrane, and also the release of stored calcium in the endoplasmic reticulum. So we have all this calcium now in the cell, and the calcium can act as a secondary messenger binding calmodulin, activating enzymes. Okay, great. So this experiment, this paper, asked the question, is calcium needed for cortisol? Is it actually necessary for calcium to be in there, or can we find some way around it? So it basically asks if we can do this whole pathway, if we can stimulate uh, the um, if we can we try to release cortisol using uh, angiotensin 2 and ACTH, but inhibit the influx of calcium from the extracellular fluid. So to do that, they would have to get antagonists of calcium channels, and it was only recently that those antagonists have become available. And there were two different ones because there were two different types of calcium channels that were studied in this paper. So there was the L-type channel. The L-type calcium channel is the L comes from long-lasting, and those calcium channels, once they're activated, will stay open for an extended period of time relatively, and will let a lot of calcium through. The antagonist to those channels is a chemical called nifedipine. Alternatively, there are T-type channels, and those are quick release or delayed release channels. So when they are activated, they stay open for a relatively shorter period of time than L-type channels do. The antagonist of T-type channels is a different chemical called TTAP2. So this paper had just a million figures, so I'm just going to pick out two of them. This one counts as one. So here we have TTAP2. They are going to add TTAP2 into a AFZ cell and see if cortisol is released. Because if cortisol is released, then it can show that calcium is not needed. If calcium is inhibited, then it shows that calcium is needed. So the control is just the cell by itself. As expected, it shouldn't release any calcium or any cortisol, and it doesn't. And then they added TTAP2 to just the cell by itself, and of course, no calcium is released. After adding ACTH, then as expected, again, cortisol is released. Then, after adding ACTH and increasing concentrations of TTAP2, they find that the cortisol is actually decreased. So this shows that if you can block the calcium channels, then you can inhibit cortisol production. And the bottom figure there is 24 hours later. They looked at the cells, found similar results. So that backs them up too. So the previous one was for the T-type channels. This is for the L-type channels with nifedipine. So nifedipine found really similar results. If you just have the cell by itself as a control, then you should get nothing. So that's the negative control. The positive control is if you add ACTH, you get a lot of cortisol being released. But after you add nifedipine in increasing amounts, then you see a decrease in the amount of cortisol released. So this shows that both the L-type and the T-type calcium channels are needed for calcium release. So here are my citations, and thank you for listening.